on turbidity, pH and microbial presence in water. To study the pH of water, using pH paper. Materials required: water samples from a pond, bore well, tap water and backwater, droppers, pH paper strips and a tile. Procedure: take a pH paper strip and place 4 strips on the tile. Using a dropper, take some pond water from the beaker. Put 1 to 2 drops of solution on the first pH strip on the tile. Using fresh droppers, do the same procedure for borewell water, tap water and backwater. Wait for some time for the pH paper to dry. Note the color and compare with the color chart given on the broad range indicator paper and get a rough estimate of pH of the sample solutions. We can observe that tap water has pH 6 and borewell water, backwater and pond water have pH 8. Using the universal indicator solution. Materials required: water samples from a pond, bore well, tap water and backwater, dropper and universal pH indicator solution. Using a dropper, take some universal indicator solution. Put 5 drops of indicator solution into the beakers that contain water samples from the pond, bore well, tap water and backwater. Note the color developed and compare it with the color chart. We can observe that tap water has pH 6 and borewell water, pond water and backwater have pH 8. To study the turbidity of water samples. Materials required: Sarkari's disc and a rope of moderate thickness. Reach the center of the pond in a small boat. Slowly immerse the Sarkari's disc into water vertically, holding the rope tightly in the hand till the black and white segments of the disc disappear. On reaching a particular depth, the disc becomes completely invisible. Length of the rope when the disc disappears, say A is equal to 95 centimeters. Slowly pull up the disc till the black and white segments of the disc just reappear. Length of the rope where it just reappears, say B is equal to 85 centimeters. Find out the mean length, say X centimeters of the rope. X is equal to A plus B by 2, that is 95 plus 85 by 2 is equal to 90 centimeters. The value of X represents the depth of the photic zone up to which sunlight penetrates in the water body and photosynthesis takes place. Precaution: Students are advised to perform this experiment under the strict supervision of a teacher to prevent accidents, including danger of drowning. Measurement of turbidity using the measuring cylinder. Materials required: water samples from pond water, backwater, and 250 milliliters measuring cylinders of the same height. Take a beaker containing pond water and transfer 250 milliliters of water sample in the measuring cylinder. Repeat the same procedure for backwater in a measuring cylinder of the same volume and height as used for the pond water. Leave them undisturbed overnight. Observe the amount of sediment settled at the bottom of each cylinder. Observation: turbidity is a measure of amount of suspended particles in the water column. Pond water has fewer suspended particles, so it has low turbidity level, and backwater has more suspended particles, so it has high turbidity level. To analyze living organisms in a water sample. Materials required: filter paper, cover slip, glass slide, needle, dropper, test tube, water sample from the pond, compound microscope. Take a beaker containing pond water and pour it into a test tube. Leave the water sample undisturbed for 48 to 72 hours. Decant the clear water, leaving concentrated sediment at the bottom of the test tube. Using a dropper, take some pond water sediment from the test tube. Drop water to the center of a clean glass slide. Take a cover slip and place it gently with the aid of a needle. Remove the extra water using a filter paper. Place this glass slide on the stage of the compound microscope and view it. We can observe large number of different organisms in a pond water sample.